In neoplasia, neo means new and plasia means growth. So, simply the name neoplasia means new growth. Although all physicians know what they mean when they use the word neoplasia, it has been difficult to develop an accurate definition. Before the development of molecular biology, British oncologist Willis made a close definition. A neoplasm is an abnormal mass of tissue, growth of which exceeds and is uncoordinated with normal tissues in the body and persists in the same excessive manner when the stimulus evoking the change is removed. Hope you can understand this definition. The current definition implies that a neoplasm is a disorder of cell growth that is triggered by a series of acquired mutations affecting a single cell and its clonal progeny. Let's try to understand what this means in the next slide. Any neoplasm in the body arises from a single cell that has undergone a genetic mutation. Therefore, neoplasms are clonal in nature, meaning that all the cells in a neoplasm consist of the same genetic composition. These mutations give the neoplastic cells a survival and growth advantage, resulting in abnormal, excessive proliferation and producing an abnormal mass of tissue called a neoplasm. Proliferation of these abnormal cells is independent of physiological growth stimuli of the body. This is called the autonomous nature of neoplasms. However, neoplasms are dependent on the body for oxygen and nutrients. Tumor was the term originally applied to the swelling caused by inflammation. However, nowadays it is considered to be a synonym for the word neoplasm. And the study of tumors is known as oncology. Tumors could be either benign or malignant. A tumor is said to be benign when its gross and microscopic appearances are relatively innocent, implying that the tumor will remain localized and not spread to other sites. Benign tumors are easily manageable with surgical removal. So, the patient generally survives. However, in some situations, benign tumors may cause significant morbidity and sometimes even death. Malignant tumors are collectively referred to as cancers. They can invade and destroy adjacent structures and spread to distant sites, which is known as metastasis. Malignant tumors are extremely fatal over benign tumors, causing significant morbidity and mortality. I will discuss about these in more detail in the following sections. Both benign and malignant tumors are composed of two essential components. Tumor parenchyma, which consists of proliferating monoclonal neoplastic cells, and reactive stroma, which consists of connective tissue, blood vessels, and cells of innate and adaptive immune system, especially macrophages and lymphocytes. The classification and behavior of tumors is based on the parenchymal component. Alternatively, growth and spread of the tumor is critically dependent on the stromal tissue component. In some tumors, stromal support is scant. Thus, the tumor looks soft and fleshy. And in some tumors, stromal support is high with abundant collagen. This is called desmoplasia. Some tumors of the breast show desmoplasia. Now we know some basic facts about tumors. And next, let's discuss about the nomenclature of tumors. Let's take benign tumors first. Most of the time, benign tumors are designated by attaching the suffix -oma to the name of the cell type from which the tumor originates. Tumors of mesenchymal origin generally follow this rule. For example, benign tumor of fibrous tissue is called fibroma. Benign tumor of cartilage tissue is called chondroma. Benign tumor of smooth muscle tissue is called leiomyoma. And benign tumor of skeletal muscle tissue is called rhabdomyoma. Nomenclature of benign epithelial neoplasms is somewhat complex than others. They are classified in several ways. Maybe according to their cells of origin. Or maybe according to their microscopic pattern and architecture. Benign epithelial neoplasms derived from glandular epithelium are called adenomas. One of the most common example is pituitary adenoma. Benign epithelial neoplasms producing microscopically or macroscopically visible, finger-like projections from epithelial surfaces are called papillomas. And benign epithelial neoplasms that produce cystic masses are called cystadenomas. Commonest example is cystadenoma of the ovary. Benign epithelial neoplasms producing papillary patterns that protrude into a cystic space are called papillary cystadenomas. Commonly seen in the parotid gland. There is another term in oncology called a polyp. A polyp could be either a benign or malignant tumor. It is a macroscopically visible projection over a mucosal surface. Polyps are commonly seen in the colon and uterine endometrium. The nomenclature of malignant tumors is not much different from benign tumors. Malignant tumors arising in mesenchymal tissues are called sarcomas. For example, malignant tumors of fibrous tissue are called fibrosarcomas. Malignant tumors of cartilage tissue are called chondrosarcomas. Malignant tumors of smooth muscle tissue are called leiomyosarcomas. And malignant tumors of skeletal muscle tissue are called rhabdomyosarcomas. Malignant tumors arising from blood-forming cells are called leukemias or lymphomas. And malignant tumors of epithelial cell origin are called carcinomas. For example, squamous cell carcinoma is the malignant tumor arising in stratified squamous epithelium. Adenocarcinoma is the malignant tumor arising in glandular epithelium. In addition, there are also some tumors are named after benign-sounding terms. Like lymphoma, which is a malignant tumor, but sounds like a benign one. Similarly, malignant tumor of plasma cells is called myeloma. Malignant tumor of melanocytes is called melanoma. And malignant tumor of mesothelium is called mesothelioma. In most benign and malignant tumors, all the parenchymal cells belong to a single type of cells. For example, in squamous cell carcinoma, all the parenchymal cells in the neoplasm are stratified squamous cells. However, there are a group of neoplasms called mixed tumors that contain different types of cells. But remember, the genetic composition of these cells is all the same in every cell. In other words, their clonal nature is preserved. When the tumor grows, cells differentiate along several pathways. Therefore, cells in the tumor will have several types of morphologies. For example, mixed tumor of salivary gland contains epithelial cells, cartilage, and bone. There are some tumors that have cells representing more than a single germ cell layer. These are called teratomas. This type of tumors develop as a result of a mutation in a totipotent stem cell. For example, cystic teratoma of the ovary contains teeth, hair, bone, cartilage, and gut epithelium. There is another type of benign tumors called hamartomas, which are disorganized but benign masses composed of cells indigenous to the involved site, such as pulmonary chondroid hamartoma. And choristoma is the presence of normal tissue in abnormal places. For example, normally organized pancreatic tissue may present in the submucosa of stomach, duodenum, or small intestine. I have taken this table from Robin's textbook of pathology. It contains the names of most common benign and malignant tumors. If you are interested, pause the video and quickly go through it. Now let's discuss about the differences between benign and malignant tumors. 4 distinct features of tumors are taken into consideration: differentiation and anaplasia, rate of growth, local invasion, and distant metastasis. First, let's discuss about differentiation and anaplasia. Differentiation is the extent to which neoplastic cells resemble the normal cells, both morphologically and functionally. In other words, are the tumor cells same as the normal cells or not? Well-differentiated cells resemble the normal cells of the tissue from which the tumor has originated. The word anaplasia means lack of differentiation. And anaplasia is considered to be a hallmark in malignancy. In general, benign neoplasms are well-differentiated. Malignant neoplasms may range from well-differentiated to undifferentiated. Anaplasia is marked by a number of morphological features. Poorly differentiated or anaplastic cells show pleomorphism, meaning that cells within the same tumor are not uniform but range from undifferentiated smaller cells to well-differentiated normal cells. And sometimes, tumor giant cells may also present. Another feature of anaplasia is abnormal nuclear morphology. Nucleus of anaplastic cells becomes larger compared to normal cells. The nuclear cytoplasmic ratio of a normal cell is around 1:4 or 1:6. But in anaplastic cells, it becomes closer to 1:1. In addition, nucleus becomes irregular in shape. This is due to clumping of chromatin along the nuclear membrane. And nucleolus also becomes enlarged. The nucleus looks darker due to abundant chromatin. Anaplastic cells also show loss of polarity, meaning that they are arranged in an unorganized manner. Normal cells in a tissue are arranged in a very organized manner, as you can see in this picture. But anaplastic cells do not have this property, as you can see in this picture. Presence of mitosis is another feature of anaplasia. In undifferentiated tumors, many cells are in mitosis, indicating the high proliferative activity. However, presence of mitosis does not necessarily indicate that the tumor is malignant, because mitosis is present even in normal tissues with rapid turnover, such as epithelia. However, presence of atypical, bizarre mitotic figures such as multipolar spindles may indicate malignancy. Rate of growth of benign and malignant tumors is determined by three factors: doubling time of tumor cells, fraction of cells that have the ability to replicate, and the rate at which tumor cells die and shed. Fast-growing tumors have a high cell turnover. Their proliferation and apoptotic rates are also high. In general, benign tumors have a low rate of growth. Malignant tumors have a high rate of growth. And most often, malignant tumors contain areas of necrosis due to this high rate of proliferation, caused by inadequate oxygen supply, as well as by compression on the tumor cells by the growing tumor itself. Local invasion is the ability of tumors to destroy adjacent tissues and replace them with neoplastic cells. Benign tumors lack the ability to invade, infiltrate, or metastasize. As benign tumors grow slowly, they develop a fibrous capsule around them. Capsule separates the benign neoplasm from host tissue. Thus, the tumor will not spread to other areas. Because of this, benign neoplasms are easily palpable and movable. In contrast, growth of malignant tumors is associated with invasion, infiltration, and destruction of adjacent tissues. They cannot be differentiated from normal tissue, and they are hardly movable. And sometimes, penetration of adjacent tissues may result in formation of fistulae. Benign and malignant tumors also differ in their ability to metastasize. Metastases are secondary tumor implants that are discontinuous with the primary tumor and located in a remote tissue. Malignant tumors have the ability to metastasize, but benign tumors do not. Now let's discuss a bit more about cancer. All cancers display 8 fundamental changes in cell physiology. These are called hallmarks of cancer. Let's discuss about them one by one in detail. First one is self-sufficiency in growth signals. Tumor cells have the capacity to proliferate without external stimuli as a consequence of oncogene activation. In our normal cells, we have a type of genes called proto-oncogenes. The function of these genes is to produce proteins that regulate normal cell proliferation and differentiation. In cancer, however, proto-oncogenes undergo mutations to produce oncogenes. As mutations upregulate the function of oncogenes, it is called oncogene activation. Activated oncogenes produce oncoproteins. These oncoproteins have the ability to promote cell growth in the absence of normal growth-promoting signals. Second one is insensitivity to growth inhibitory signals. Our normal cells also have a type of genes called tumor suppressor genes, which encode proteins that regulate cell cycle checkpoints. When a DNA-damaged cell undergoes division, these proteins will halt its proliferation and drive the cell to initiate DNA repair. If DNA repair is not possible, they induce cell death by apoptosis. In cancer, however, these tumor suppressor genes are inactivated, resulting in uncontrolled proliferation of mutated cells. Examples for tumor suppressor genes include retinoblastoma gene, p53 gene, also known as guardian of the genome, APC gene, WT gene, and BRCA1 and 2 genes. I encourage you to find specific types of cancers associated with these genes as an activity. Third hallmark of cancer is altered cellular metabolism. In general, glucose uptake of malignant tumor cells is high. However, they prefer lactic acid fermentation over oxidative phosphorylation for ATP generation. This is called the Warburg effect. If glucose has undergone oxidative phosphorylation, it produces more energy, but all the carbon molecules in glucose are lost as carbon dioxide. And, even the energy production is less. Lactic acid fermentation rapidly yields metabolic intermediates that are required for the synthesis of macromolecules, essential for the growing tumor. This is the main reason that tumor cells prefer lactic acid fermentation as their primary energy production pathway. Next one is evasion of apoptosis. We already know that when a cell with genetic damage tries to undergo cell division, it is recognized by p53 genes and halt its proliferation. Then the cell is driven towards DNA repair, and if it is not possible, the cell will undergo death by apoptosis. This apoptotic process is regulated by another type of genes called apoptosis-regulating genes. In cancer, however, these genes are also damaged. So, there will be accumulation of cells with genetic damage, which facilitates development of malignancy. Fifth hallmark of cancer is their limitless replicative potential, also referred to as immortality of cancer cells. Normal cells in our body can divide only up to 60 to 70 cycles. But cancer cells can divide limitlessly. 3 closely related factors are responsible for the immortality of cancer cells. One is evasion of senescence. With cellular aging, the ability of division in our normal cells declines. And the cells become senescent. This is thought to be due to the upregulation of p53 gene activity with cellular aging. However, in cancer, p53 gene function is impaired, as we discussed earlier. So, the cells do not reach the state of senescence. Second mechanism is evasion of mitotic crisis. In our chromosomal ends, we have specific areas called telomeres. These are specific DNA sequences that bind with protective proteins. The main function of telomeres is to prevent enzymatic degradation of chromosomal ends and to maintain genomic stability. In normal cells, telomeres shorten with each division. And finally, they undergo death due to destruction of DNA. This is called mitotic crisis. However, some cells have the ability to resynthesize telomeres as they have the enzyme telomerase. Cancer cells also have this ability. By doing so, they can evade mitotic crisis. Third mechanism is ability of self-renewal. This means that with each division of a stem cell in neoplasia, one daughter cell undergoes differentiation and becomes a mature cell. And one daughter cell remains a stem cell. So, with time there will be more and more stem cells in the neoplasm, ensuring that the tumor will not run out of stem cells, which are essential to increase the size of the tumor. Sixth hallmark of cancer is sustained angiogenesis. Formation of new blood vessels is called angiogenesis. Like all other cells, cancer cells also depend on the body for oxygen and nutrients. So, they need a good blood supply to maintain their proliferation rate. In normal tissues, angiogenesis is tightly regulated by a balance between angiogenesis promoting and inhibiting factors. In cancer, however, this balance is lost. Maybe due to upregulation of angiogenesis promoting factors. Or maybe due to downregulation of angiogenesis inhibiting factors. Or maybe due to both. These factors are secreted by tumor cells, macrophages, and other stromal cells. Examples for angiogenesis promoters include vascular endothelial growth factor and basic fibroblast growth factor. Examples for angiogenesis inhibitors include angiostatin and endostatin. Seventh hallmark of cancer is ability to invade and metastasize. In other words, spreading of the tumor to other sites. I will discuss about this in more detail towards the end of this video. And the final one is ability to invade host immune response. Our immune system constantly scans for malignant cells and destroy them. This is known as tumor immune surveillance. Due to mutations, malignant tumor cells express abnormal antigens. Our immune system recognizes these as foreign agents. So, they eliminate these cells by destroying them. Main cells involved in tumor immune surveillance are T cells, macrophages, and natural killer cells. In immunocompromised patients, tumor surveillance is poor. So, they are more susceptible to get cancers. Some tumor cells have the ability to escape the host immune response by several mechanisms. Tumors produce large amounts of antigen-negative cells and small amounts of antigenic tumor cells. Antigen-negative cells cannot be recognized by the immune cells. So, they can easily escape the immune response. And some tumor cells secrete cytokines to inhibit immune cells. Some tumor cells have the ability to mask their antigens. For example, some cells secrete mucopolysaccharides to cover their antigens. And some tumor cells bind to activated platelets in blood and hide their antigens. Contact-dependent killing of immune cells by the tumor cells is another mechanism. And some tumors recognize cytokines as growth factors. So, instead of being killed, they undergo proliferation using cytokines. Now let's discuss about the spread of malignant tumors. This topic is extremely important in oncology because invasion of adjacent tissues and metastasis are the major causes of cancer-related morbidity and mortality. Metastatic cascade is divided into two phases: invasion of extracellular matrix and vascular dissemination, homing of tumor cells, and colonization. First, let's discuss about invasion of extracellular matrix. This is a four-step process. At first, tumor cells detach from each other due to loosening up of cell-to-cell interactions. Then, they degrade the extracellular matrix by releasing proteases. In the third step, tumor cells attach to novel extracellular matrix components. And the final step is migration and invasion of tumor cells. In normal tissues, cells are tightly bound together by transmembrane glycoproteins belonging to the family of cadherins. However, during invasion of malignant tumors, cadherin production is down-regulated. resulting in detachment of cancer cells from each other, as indicated by the yellow circle in this picture. In the second step, tumor cells secrete proteolytic enzymes to degrade extracellular matrix proteins. They also induce surrounding fibroblasts and inflammatory cells to secrete these enzymes. Main proteolytic enzymes include matrix metalloproteinases, cathepsin D, plasminogen activator, and collagenases. In the third step, tumor cells dissociate from the extracellular matrix proteins and bind new extracellular matrix components. In normal tissues, cells are bound to extracellular matrix proteins like collagen and laminin through integrin receptors. This binding helps to keep all the cells in a polarized manner. In cancer cells, these receptors are down-regulated, resulting in detachment of tumor cells from extracellular matrix proteins and binding with new extracellular matrix components. If normal cells are mobilized from the extracellular matrix like this, they undergo death by apoptosis. But, as tumor cells can escape apoptosis, they survive after detachment from ECM proteins. Migration and invasion of tumor cells is a multi-step process involving many types of receptors and signaling molecules. Cleavage products of matrix proteins and growth factors such as insulin-like growth factor act as chemotactic agents and facilitate the amoeboid movement of cancer cells. Finally, they make their way into the vascular system in order to metastasize. This is called intravasation. Next phase of metastatic cascade is vascular dissemination, homing of tumor cells, and colonization. Metastasis is the process of producing tumor deposits at a site distant to the initial mass. And these secondary deposits are discontinuous with the primary tumor. Metastasis occurs via three pathways: seeding of body cavities and surfaces, lymphatic spread, and hematogenous spread. Seeding of body cavities and surfaces occurs when the tumor directly penetrates into a natural open field. These areas lack physical barriers. One common example is peritoneal cavity. Lymphatic spread is the most common way for the spread of carcinomas. But some sarcomas may also disseminate via lymphatics. Hematogenous spread is the most common way for the spread of sarcomas. Arteries are usually resistant to invasion by tumors because they have a thick wall and higher pressure inside. So, most tumors prefer veins over arteries. Once in the circulation after intravasation, tumor cells are vulnerable to destruction by several mechanisms, including mechanical shear stress, innate and adaptive immune defenses, and apoptosis induced by mobilization from the extracellular matrix. So, tumor cells tend to clump together in the circulation as an adaptive response to escape these mechanisms. They also bind activated platelets and coagulation factors. This whole structure is called a tumor embolus. Formation of tumor emboli helps tumor cells to withstand shear stresses and escape the immune response in the circulation. Once a favorable soil is reached, tumor cells then extravasate from the circulation in order to produce metastatic deposits. The site of metastasis is determined by natural pathway of venous and lymphatic drainage and organ trophism of tumor cells. Finally, let's discuss about some common clinical consequences due to the spread of malignant tumors. First, let's take local effects. These effects can be categorized into three groups: effects due to the tumor mass, effects due to loco-regional spread, and local hormonal effects. These effects of local invasion are dependent upon the site of the tumor. Compression on the normal tissue and adjacent structures by the tumor may cause loss of function of the affected area. For example, tumors in brain tissue may cause certain neurological defects and herniation of some part of the brain. As tumors have a high proliferation rate, some cells may not receive adequate oxygen to maintain their functions. So, they may undergo necrosis. This will result in ulceration of the affected area. For example, in colon cancer, mucosa of the colon frequently get ulcerated due to necrosis. Some tumors act as a nidus for infection. For example, some people develop bacterial pneumonia in lung cancer. When the tumor involves a lumen of an organ, it may cause luminal obstruction. For example, in colon cancer, lumen of the colon gets obstructed due to the tumor mass. Another manifestation of malignant tumors is perforation of the organs. For example, malignant perforation of gastric ulcers, and this may result in peritonitis ultimately. Local hormonal effects occur when the tumor is a hormone-secreting one. This may result in excess hormone levels in blood. Effects due to loco-regional spread may occur when the tumor erodes into surrounding structures like veins, nerves, and body cavities. For example, invasion of veins may cause varicocele in patients with renal cancer. Invasion of nerves may cause nerve palsies. And invasion of adjacent body cavities may cause effusions. Most of the time, cancers cause systemic manifestations as well. Cancer cachexia is one of the most common manifestations in cancer. It occurs in about 50% patients with cancer. It is characterized by the equal loss of both fat and lean muscle, elevated basal metabolic rate, and evidence of systemic infection. Tumor necrosis factor is the main mediator responsible for cancer cachexia. Inhibitory effect of tumor necrosis factor on the hypothalamus causes anorexia. And many patients will have increased levels of acute phase proteins. Paraneoplastic syndromes are a group of disorders that are triggered by an abnormal immune response against malignant tumors. Occur in about 10 to 15% of patients with cancer. Patients with paraneoplastic syndromes will have a wide variety of symptoms and diseases, including hypercalcemia, hypoglycemia, polycythemia, disseminated intravascular coagulation, finger clubbing, Cushing's syndrome, acanthosis nigricans, and nephritic syndrome. Okay. That's all I wish to discuss in this video. I have covered most of the learning material required for general pathology on neoplasia. But do not forget to read your textbooks. There is another topic in neoplasia, the molecular basis of cancer. I will discuss about it in another video.